Welcome to the Deep Dive, where we uncover fascinating scientific discoveries just for you. Today we're taking a deep dive into a research paper from the University of Pennsylvania.
Oh, very cool.
It's all about a protein called VCP and how it keeps the nucleus of our cells tidy. I'm sure many of our listeners are familiar with VCP.
Yes, VCP is a fascinating protein. You see, the nucleus is like the control center of the cell where all the important genetic information is stored.
Right.
It's crucial to keep this area free of clutter.
Absolutely. It's like trying to run a company with a messy office. Nothing will get done.
Precisely. And this research focuses on a serious genetic disease called multisystem proteinopathy, or MSP for short.
MSP.
Yes, MSP impacts multiple body systems,
Oh, wow.
including muscles, bones, and even the brain.
The name itself sounds pretty intense. What causes this multi-system issue?
Well, researchers have found that mutations in the VCP gene are a major culprit.
I see.
This research explores the connection between these VCP mutations and a buildup of abnormal proteins inside the nucleus. Interestingly, this buildup was observed in tissue samples from patients with 3 different conditions linked to MSP, a type of dementia called FTLD-TDP, which affects the brain, IBM, which affects muscles, and Paget's disease of bone, which impacts bone formation.
So 3 seemingly different conditions all share a common thread, this buildup of abnormal proteins in the nucleus.
Exactly.
Is this where VCP comes into play?
Yes. VCP usually acts like a foreman for the cell's cleanup crew. It helps break down and recycle old or damaged proteins, preventing them from forming harmful clumps.
So it tags these old proteins with a signal that basically says, take me out. How does that signal work?
Think of it like a garbage sticker.
Okay.
You put a sticker on something you want the garbage collectors to take away. In the cell, that sticker is a molecule called ubiquitin. VCP helps attach ubiquitin to proteins that need to be removed.
And what happens in people with MSP? Is their cellular garbage collection system breaking down?
That's exactly what the researchers wanted to find out. They created special cells with the same VCP mutations found in MSP patients. Oh, wow.
What they found is that these cells had much lower levels of VCP inside the nucleus.
So the foreman is missing from the control center. No wonder things are getting messy. But how did they actually show that this lack of VCP was causing the protein buildup?
They focused on a protein called TDP43, which is known to form harmful aggregates in FTLD-TDP.
Right.
Remember, that's a dementia linked to MSP.
Yes.
They used a version of TDP43 that's prone to clumping, and by blocking the cell's recycling center, they intentionally created a situation where these clumps would build up.
Like purposely overloading the garbage disposal to see if it could handle it.
Yes, exactly.
And I'm guessing it couldn't.
Not entirely. They then used a drug called CB5083, which specifically inhibits VCP.
Okay.
Think of it like putting the foreman on vacation. When they added CB5083 to cells already struggling to clear the TDP43 aggregates, the situation got even worse. This confirmed that VCP's activity is crucial for breaking down these protein clumps, even inside the nucleus. So even in the control center, VCP plays a vital role in keeping things organized. But this research isn't just about understanding the problem, right?
No.
The goal is to find a solution.
Absolutely. And that's where things get really exciting.
Okay, I'm all ears. Tell me more. So they knew VCP was important and that boosting its activity could potentially help clear out those protein clumps. What did they do next?
Well, they dove into a massive database of chemical compounds, looking for anything that could increase VCP activity.
Wow.
And they found not 1, but 4 potential candidates.
Really?
These compounds acted as VCP activators. So instead of inhibiting VCP, like that CB5083 drug we talked about earlier,
Right.
these new compounds actually boosted its performance.
That's amazing. It's like giving the cleanup crew extra energy drinks.
Yeah.
But how did they know these activators would actually work in the context of MSP?
They were very thorough.
Okay.
First, they tested the compounds on VCP in a test tube, just to make sure they were actually increasing its activity.
Right.
Then they moved on to cells, adding the compounds to those struggling with those stubborn TDP43 aggregates. Remember, those are the clumps that were building up in the nucleus.
So they started with a controlled environment and then moved to a more realistic model. Did any of these compounds stand out?
One compound in particular emerged as a star player. It's called UP109.
UP109.
UP109 was incredibly effective at boosting VCP activity in the lab. It significantly improved VCP's ability to process ATP, which is the cell's main energy source.
So it's like UP109 supercharges VCP, giving it the energy it needs to tackle those protein clumps. But how does it actually do that?
Well, UP109 seems to specifically target the D2 ATPase domain of VCP. Now remember, VCP has two of these ATPase domains, D1 and D2. You can think of them as the engines that power VCP's protein unfolding abilities.
So UP109 is specifically targeting one of the engines. Why is that important?
Well, the D1 domain is responsible for initially grabbing onto the protein that needs to be unfolded, but it's the D2 domain that does the heavy lifting. It provides the force to actually pull the protein through VCP and unfold it.
Okay, so UP109 is basically giving a boost to the engine that's responsible for the most challenging part of the cleanup process.
Yeah.
No wonder it was so effective at clearing out those TDP-43 clumps.
Exactly. And the research showed that UP109 not only increased the speed at which VCP could unfold proteins, but it also improved its efficiency in using ATP. It's like giving the cleanup crew better tools and teaching them how to use their energy more effectively.
This is all incredibly promising, especially for those suffering from MSP. But I know we can't get ahead of ourselves. What are the next steps in this research? What needs to happen before UP109 can be considered as a potential treatment for humans?
Well, first and foremost, we need to see how UP109 behaves in a more complex environment. Remember, these initial experiments were all done in cells, either in test tubes or petri dishes.
Right, so the next step would be to test it in animal models.
Exactly. We need to make sure that UP109 is safe and effective in a living organism before we can even think about human trials. And even if it passes those tests, there's still a long road ahead.
Of course. There's always the challenge of translating promising results from the lab into real-world treatments. But even so, this research gives us a reason to be optimistic.
Absolutely. It's a significant step forward in our understanding of MSP, and it highlights the potential of VCP as a therapeutic target. Who would have thought that a protein responsible for cellular housekeeping could hold the key to treating such a complex disease?
It's amazing what we're learning about the inner workings of our cells.
Mhm.
But I have to admit, I'm still struggling to visualize how VCP actually works. We've talked about ATPase domains and unfolding proteins, but I need a clearer picture.
I understand. It's one thing to hear about these processes, but quite another to actually picture them happening. Maybe a visual analogy would help.
An analogy, I love analogies.
Okay, imagine a construction site.
Okay,
There's a lot of debris that needs to be removed. Old lumber, broken concrete, twisted metal, that sort of thing.
okay, I can picture that.
Now imagine VCP as a powerful crane responsible for moving all that heavy debris.
So the debris represents the misfolded proteins that need to be cleared out.
Exactly. Now for the crane to operate, it needs fuel, right?
Of course, otherwise it can't lift anything.
Well, in this case, the fuel is ATP. It provides the energy that VCP needs to function.
And the ATPase domains we talked about earlier, those are like the engines of the crane, using the ATP fuel to power the crane's movement.
Precisely. Now, the D1 domain is like the claw of the crane, grabbing onto the debris, while the D2 domain is the powerful motor that lifts and moves the arm, essentially unfolding the protein as it's being pulled through VCP.
Okay, I think I'm starting to get it. So it's not just about breaking down the proteins, it's about unfolding them so they can be properly recycled or disposed of.
Exactly. And when VCP is working correctly, it ensures that the construction site stays clean and organized. But when there are mutations in VCP, like in MSP, the crane is either low on fuel or the motor isn't working properly, leading to a buildup of debris.
And that's where UP109 comes in. It's like giving the crane a supercharged engine, allowing it to work more efficiently and clear out that debris, even if there are some mechanical issues.
That's a great way to put it. This analogy really helps us appreciate the complexity and importance of VCP's role in maintaining cellular health.
It definitely does. And it makes the potential of UP109 as a treatment for MSP even more exciting. But before we get too carried away with our construction site metaphors, let's shift our focus to the broader implications of this research. That crane analogy was really helpful. It's amazing how such a simple image can clarify a complex process. But I'm curious, this research focused specifically on MSP.
Right.
But aren't there other diseases linked to protein misfolding?
Oh, absolutely. You're absolutely right. Protein misfolding is a hallmark of many neurodegenerative diseases.
Oh, wow.
Alzheimer's, Parkinson's, Huntington's disease, to name a few.
So if UP109 can enhance VCP's ability to clear out those TDP-43 clumps in MSP, could it potentially work for other types of misfolded proteins as well?
That's a great question. It's a question that researchers are actively investigating. The idea of targeting VCP to address protein misfolding in various diseases is incredibly exciting. However, we need to exercise caution. Each protein has a unique structure,
Right.
and the way it misfolds and aggregates can vary. What works for one protein might not work for another.
So it's not a one-size-fits-all solution.
Exactly.
But even so, this research opens up a new avenue for exploration.
Exactly. It emphasizes the importance of understanding the specific mechanisms behind protein misfolding in each disease. VCP might play a key role in some cases, but not in others. More research is needed to determine its therapeutic potential across different conditions.
So while there's still much to learn, this research gives us a reason to be hopeful. It's fascinating to think that a protein primarily associated with cellular housekeeping could hold the key to treating such devastating diseases.
Indeed, this deep dive has revealed the intricate connection between VCP, protein misfolding, and MSP. It has also highlighted the potential of UP109 as a therapeutic agent. While further research is needed to confirm its safety and efficacy in humans, the results so far are very promising.
And beyond MSP, this research has broadened our understanding of VCP's role in maintaining cellular health. Who knew a cellular foreman could be so crucial?
It just goes to show that even the smallest players in our cells can have a profound impact on our overall well-being. The more we learn about these intricate processes, the better equipped we'll be to develop effective treatments for a wide range of diseases.
This has been a truly enlightening deep dive. We've gone from the control center of the cell to a bustling construction site, uncovering fascinating insights along the way.
I've enjoyed our exploration as well. It's always rewarding to delve into cutting-edge research and discuss its implications.
And I especially appreciate how you helped me visualize the role of ECP. That crane analogy really brought it all together.
I'm glad it was helpful. Analogies can be powerful tools for understanding complex scientific concepts.
Well, I'm already looking forward to our next deep dive. Who knows what fascinating discoveries await us?
Indeed. The world of science is full of endless wonders. There's always something new to explore and understand.
That's the beauty of it. And we'll be here to guide you through those explorations, uncovering the mysteries of science one deep dive at a time.
Until next time.